For this week, I am going to spend most of the time on Lab 5. We're not going to spend a whole lot of time on Lab 3 because the organelle stuff we've gone over in lecture. So what you do need to know for lab, like for the practical and for the quiz in here from Lab 3 is basically table 3.1. So that's the part I'm not going over today. That's just the organelles and their functions. We did that in lecture. So focus on that. I'm not going to ask you to like identify the different shapes of an organelle on a drawing of a cell. You don't have to worry about that. And then the other aspect of Lab 3 is mitosis. And I'm going to go over mitosis fairly quickly, assuming most people have kind of seen mitosis before, at least even in kind of passing. So I'm going to point out what I want you to know about mitosis for lab. I doubt we'll get to it in lecture, so this might be the only time you see it this semester. Yeah, do this. Okay. So I'm going to, like I said, the kind of basics of mitosis. So first off, you want to know why cells do mitosis, what the whole point is, and then each of the stages and the main events in each stage, not every single event. So on the next few slides, I have images of these different stages and then bullet points of what the events are that you want to be familiar with in each stage. There's really no more than 2, I don't think, for each stage. Most of them just have 1. And this PowerPoint is on Canvas already. And then the Lab 5 PowerPoint is on Canvas as well. You're definitely going to want to review the Lab 5 PowerPoint. There's a lot of tissues and pictures of tissues, which can be kind of difficult to distinguish. So both of those are already up, and the quiz is already up for this week. So you can get that out of the way whenever you want between now and next Tuesday. Okay, so first, mitosis quickly. The whole point of mitosis is for one cell to become 2 cells. So basically a cell is cloning itself. It's an exact copy of the cell. So same genetic information, and that genetic material needs to be present in both of the resulting cells. So the genetic information gets copied before we get into mitosis. So I'm not really talking about the DNA replication aspect of this, but we'll imagine there's 2 copies of the DNA in the cell that's about to split that we're going to look at. And one copy is going to go into one resulting cell. The other copy will go into the other cell. These are the 4 stages of mitosis: prophase, metaphase, anaphase, and telophase. That's the order that they happen in. So you want to know the order as well as the names, and then be able to identify them based on pictures or the events that happen, which I'm about to go into. You may have learned prometaphase, but I just ignore prometaphase because I think it complicates things. So we're not going to talk about prometaphase. So first we have prophase. In prophase, the cell is really at the beginning steps of starting to split. So the DNA is condensing into chromosomes. So before, it was just this kind of cloudy, hazy nucleus in the middle. You don't see any chromosomes yet. In this cell, the chromosomes are starting to condense, so you can kind of see little individual lines. That's the indicator that prophase is happening. And the nuclear membrane, you may or may not be able to see here at this point, but look at it on Canvas later. It's starting to kind of fragment and break down. So those are the 2 events in prophase that you want to be familiar with. What am I doing? The second stage is metaphase. Metaphase is easy. The chromosomes just line up along the midline of the cell. So metaphase, middle. Super easy to remember. That's the only event that happens for our purposes during metaphase. So you see the chromosomes lining up along the middle. And remember, chromosomes are kind of this X shape, right? So the left side and the right side are exact copies of each other at this point, and they're about to be pulled apart. Anaphase is where what we call sister chromatids. So a chromosome, as we think of it, is made up of 2 sister chromatids. Those chromatids get split apart and move to either end of the cell. So it's still just one cell, but the DNA, now there's going to be a copy of DNA over here and an exact copy of the DNA over here. So same DNA split onto opposite sides. That's anaphase. And then really the only other aspect you need to understand about mitosis is the telophase step, where the cell starts to kind of pinch in the middle. And eventually, after telophase, it will completely split in half. But you have the genetic material in both of the almost new cells. And the cytoplasm is starting to pinch in the middle, creating what's called a cleavage furrow. And that will eventually break into two cells. And that is the most important part of telophase. And that kind of wraps up all I want you to know about mitosis. So just a really quick review, hopefully, of what happens when a cell splits. Any questions about that or what you need to know? Okay. So that's kind of the minor, easy parts of this lab. Well, this lab session, that was its own lab. For the next part, this is lab 5. So for lab 5, we're going to get into tissues. And I haven't talked about tissues yet in lecture. So we haven't gotten there. So I'm going to spend a bit of time. Most of this lab, if you haven't figured out already, is just me talking. So today I'm just going to be talking a lot, mostly because we haven't done this yet in lecture. So I need to give you guys enough background on it so you kind of understand tissues. The handouts that I put over here are just an opportunity to draw these tissues if you find that useful. So there's a lot of different kinds of tissues, and I've broken it down by kind of main categories, more or less. So epithelial tissues on the front, and then you have in the middle different kinds of connective tissues, and on the back, the muscle and nervous tissue. It can be helpful to draw these tissues out. They can be really hard to distinguish. So I guess I should, before I jump into the tissues, start with kind of warning you guys that this is an area that can trip a lot of people up. So it's a really difficult topic to learn because it's hard to tell the difference between them when you look at the slides. There's a lot of them, and it's not particularly exciting. So there's really nothing to attach to there's really nothing to attach your understanding to with these tissues. It's a little bit tough to envision what they are and what they're doing because they're just a bunch of cells working together. So this isn't necessarily, if you feel overwhelmed by this, you're not the only one. I guess that's my way of saying just stick with it and you will learn them. You just have to really put in time with these tissue types. There's kind of no easy way to do it. So that being said, you can draw them if you like. I'm not gonna check your drawings. I'm a terrible artist and I hate drawing things. So I would never force people to draw anything. But it can be really helpful, even if no one ever sees it. If you can draw these tissues, then you understand how they're structured and how to distinguish between the different types. But that is up to you. So I'm going to run through these different tissue types. And my goal today is to give you some helpful hints as to how to identify them. So in your lab, there's lots of good pictures in your lab book for this lab. I added, some of them are up here, but I also added some different ones to give you a different view of some of them. But it also, with each of the kind of subclasses of tissues, it tells you 3 things in the lab manual. It tells you a description, a function, and a location. You don't have to, for lab, you don't have to worry about the function or the location. I really just want you to be able to, you see a slide or hear a description or read a description of a tissue and identify what that tissue is. So it's really just understanding and being able to identify the tissues in here. When it comes to lecture, we'll go over the function and where these tissues are found. So you'll need that eventually, but not quite yet and not for lab. So I'm not going to ask you that stuff on the quiz or the practical in here. Just be able to identify them. Okay. I think that's the main points. So there's 4 main categories. And the tough part about these tissues, one of the tough parts, is that there's subcategories and then kind of subcategories with some of them. So it can be helpful to outline how these are related to each other. If outlines help you, this is a great place to outline where these different tissues, how they relate to each other in outline form. So that's kind of how I'm breaking down this PowerPoint. So these are the four main categories, the big categories of tissues. You have epithelial tissues, which are closely packed cells that are all connected to a basement membrane, and I'll show you what that looks like. But they're all packed in there right next to each other, like this. But lots of closely packed cells. Connective tissue is more widely spaced. So these purple things are the cells, and then there's space in between the cells. So they're not packed up against each other. Muscle tissue is pretty distinctive. So we'll get to that. Muscle and nervous tissue are kind of the easier ones. Nervous tissue is super easy. There's only one kind, and it always kind of looks like this. Muscle tissue is fairly easy as well. The connective and epithelial tissues are the hard ones. So that's what I'm going to start with. I'm going to start with epithelial. I would say epithelial is probably the hardest to identify, followed closely by connective tissue. So if you can focus for the next 30 or so, not even 30, probably 20 minutes on this information, it will get you a long way into kind of understanding and not having to wrestle. You'll still have to wrestle somewhat, not have to wrestle as much with identifying these slides of tissues. Okay. So let's start with epithelial. And epi means on top of. So epithelial tissue, again, you don't need to know where it's found for lab, but I think it's helpful to envision, at least in this case, what the heck it's doing. So it's covering our external body surfaces. So your skin is epithelial tissue, and it's also lining internal cavities. So your digestive system has a lot of internal cavities, your intestines, your stomach, your respiratory system, your lungs, trachea, etc. All of those are internal cavities, and they're lined by epithelial tissue. Okay, epithelial tissue is broken down based on how many layers of cells there are, and then also what shape the cells are. So those are the two categorizations of epithelial tissues. As far as the number of cell layers, you can have one, or you can have more than one. So one layer of cells, it's called a simple epithelial tissue. Multiple layers of cells, it can be 2, it can be 10, whatever. It's stratified. So stratified is multiple layers. Simple is just one layer. So if we look up here, this is just one layer of cells. This is many layers of cells. And this basal surface that's noted here is that basement membrane. So the lowest layer, even if it's just one, is attached to that basement membrane. So it's all attached to something on the bottom. In addition to how many layers there are, we also have to consider what shape the cells are. So there's 3 options here. They can be squamous, which is flat. I've also heard it pronounced squamous, but I always say squamous. So it's a flattened cell. So this would be looking at the cell like a cross section, looking at the side of it. So flattened cell, that purple thing is the nucleus. The nucleus can really help you identify these different cells as well. So in a squamous cell, that nucleus is really smushed out and flattened, just like the cell. A cuboidal cell is cube-shaped, about like more or less square. And the nucleus is going to take up a really large portion of it. So it's going to look like the nucleus is most of the cell in cuboidal cells. And then columnar is column shaped, pretty straightforward, just a tall rectangle. And the nucleus is usually a little bit elongated. In cuboidal, it's a nice sphere. Columnar, you have this kind of elongate nucleus within the cell. So each type of epithelia is going to have one of each of these names with it. So you'll have simple squamous, or simple columnar, or stratified squamous, or stratified cuboidal. So you have to have the number of layers option as well as the shape. Both of those adjectives are basically going to go into the name of these epithelial tissues. And all possible combinations, so there will be 6. And I actually, on this handout, I should have put 8 circles. There's 2 that don't fit into this categorization, which makes it hard. So there should be 1 more circle on the epithelial tissue slide, sorry. Keep going my computer, changing the slide. I think this diagram is really helpful to sort of simplify those 6 different options. So on the top, you have the simple or 1 layer with each of the shapes. Simple is pretty easy. The shape is fairly obvious most of the time and easy to distinguish between the 3. When you get into the stratified, it gets a little bit more complicated. So the main issue here is that if we're looking at the squamous cells, at the bottom, they're actually kind of cube shaped, right? They're not flattened out at the bottom. So in these stratified options, you want to look at the top layer of cells for the shape. So these top cells are flattened, so that's considered squamous stratified epithelia. Cube is pretty much, cuboidal is the same no matter what layer you're looking at. Cuboidal is pretty easy, stratified or simple. And then columnar, we run into the same issue with the bottom layers. They're kind of cuboidal, but the top layer is definitely made up of column-shaped cells. So that's considered stratified columnar epithelia. So if you're looking at multiple layers of cells on top of each other, look at the top layer for the shape description. Okay. So this makes it look really simple. When you look at actual slides, it's obviously not nearly as clear as a nice drawing like this. So I'm going to run through what you'll actually be looking at and identifying with these different tissue types. These are the 3 simple options. So simple squamous, simple cuboidal, and simple columnar. The simple squamous, you can't really get a side, you can't get much of a side view of, but this is just 1 layer of flattened cells. They're not cube shaped, they're just kind of weirdly irregular. So that's going to be your simple squamous. Simple cuboidal over here. So the cuboidal, like I said, the nucleus takes up a lot of the cell. So these are all individual cells. That dark purple is the nucleus. It's taking up a massive amount of the cell. So that's a good indicator it's cuboidal, if you can't tell that the cells are kind of cube shaped. And this is simple. You see a lot of layers here, right? So this is where it gets confusing as to why it's considered simple rather than stratified. So you have to think about it as this epithelial tissue is lining these cavities. So these big white areas are some kind of cavity in the body. This would be a big cavity here. You only have 1 layer of cells on either side, and then you have another cavity, but this one is hard to see, right? So when I ask you or give you an image of this, I try to make it as clear as possible that it's 1 layer. When you see the stratified cuboidal, it'll be kind of obvious that there's 2 right next to each other up against a cavity. But this is just 1 layer of cells around each of these cavities. So that is simple cuboidal. And then simple columnar down here, pretty easy. Really tall cells with sort of ellipse-shaped nuclei. And then down here is the basement membrane, which is sort of hard to tell, but I would let you guys know that this is the basement membrane, and you only see 1 layer of cells, and they're really tall, so you know it's simple cuboidal, or sorry, simple columnar. All right, so those are our simples. Moving on to the stratified. So these are our stratified options. The two top ones are both squamous. One is just kind of more zoomed out. So this one on the upper left is kind of showing a large cross section of skin. And you'll learn these different layers when we get into the integumentary system. But imagine this kind of section up here, the epidermis area, that's what this is. So we're just kind of zoomed in. So stratified squamous can be difficult to identify because if you look over here, I guess they're not really cube shaped. There's some really abnormal shapes. They're not nice and neat like this cube down here. But I would indicate to you, focus on this area to identify this tissue because there's a lot of squished cells here. So those are the stratified squamous. So they're all layered on top of each other and they're kind of squished out into weird, somewhat flattened shapes. Okay, stratified cuboidal over here. So here we have the cavity that is lining, and now we have two layers of cells. So if you compare this to the simple cuboidal, hopefully it's pretty obvious that the simple cuboidal is one layer of cells, and this is clearly two layers of cells kind of stacked on top of each other, so that makes it stratified. And it's cuboidal because, again, these nuclei are taking up the vast majority of the cell. And then stratified columnar. Stratified columnar can be tough to distinguish from one of the weird ones I'm about to get into. But here you have, this is going to be the basement membrane right here. And then you can see these nuclei. That's kind of how you identify these cells. So you have all these nuclei lining up. That's the first layer of cells. But they're kind of cube shaped. They're definitely not columnar. So you need to look at the top layer. Those are column shaped cells. You have the somewhat elongate nuclei that are not taking up most of the cell. So you have a lot of cell area that's not taken up by the nucleus. But like I said, any picture that I give you, I'll try to make it as obvious as possible as to what these different tissue types are, assuming you know what to look for. I think this is kind of tough to tell, in my opinion. So I should probably choose a better picture for the PowerPoint. Okay, so those are the six types of epithelial tissues that fit into these nice, neat categories. These are the two that are kind of weird and don't fit into any of those categories. They're kind of in between some of them. So we have what's called pseudostratified columnar epithelium. And pseudo means kind of, so it's kind of stratified, it's kind of layered, and it's columnar. So what this is basically there's different lengths of columnar cells. So some reach all the way up and some are kind of short, but they're not layered on top of each other. It just appears like they're layered. So you see all the nuclei at differing heights. So you would kind of guess, oh, this is layered, but really, if you have a decent enough picture, you'd be able to tell that it's not one layer on the bottom and then a separate layer on top. It's actually just all these different size cells, some of which reach to the top and some of which only say reach to here. But this one doesn't have another cell directly on top of it. It's not stacked right on top of it. Still just one layer technically. And you can see it up here too. So all these nuclei are kind of at differing levels. There's not one layer of nuclei and then another layer of nuclei. So that's a good indicator that it's pseudostratified. And transitional epithelium is a type of tissue that's found in organs that can stretch and then relax. So the bladder is a great example. It has transitional epithelium in it. So when your bladder is full, it's really stretched out and the cells on top get flattened out into a squamous shape. And then when your bladder is empty, it relaxes and the cells on top become more cube shaped. So the shape actually changes. And transitional epithelium is a tough image for me to give you because you'd have to have something to compare it to. So for that reason, if I ask you about transitional epithelium, it'll probably just be in some kind of word form. Like this is a type of tissue that the cell shapes change depending on whether the organ is stretched or relaxed, something like that. But here you can kind of see there's mostly column-shaped cells and then some weird globular cells on top. Those globular cells on top are what flatten out or relax, or I guess go more cube-shaped. So those are the two weirdos that don't really fit into any of the other 6 categories. And that's all the epithelia. Like I said, I think those are the hardest. I think some people find connective tissues a little bit harder, but you know, they're kind of tied for difficulty. So that's one of the really hard ones, one of the really hard categories. Now we'll get into the next one, connective tissue. There are technically 4 subcategories, but we're ignoring the 4th one, which is blood, because we don't talk about the circulatory system in AMP1. That's in AMP2, so we're just going to ignore blood. We will talk about these other 3. Connective tissue proper, which is kind of just general connective tissue. It's a good way to think of it. And then cartilage, which I think most people are familiar with. It's in your nose, it's in your ears. It's kind of solid, but not really as solid as bone, kind of an in-between. And then there's bone. So bone is a connective tissue, and it's actually pretty easy to ID. The other 2 are a little bit more difficult, connective tissue proper and cartilage, and they each have subtypes within them. Bone is just bone, it doesn't have any subtypes. Okay, so connective tissue, I told you epithelial tissue kind of lines the external body or internal compartments. Connective tissue is far and away the most common tissue in our body and is basically sort of holding everything in place. It's connecting everything. So let's start with the connective tissue proper. There are both loose and dense types of connective tissue. There's only one kind of dense connective tissue you need to know. So I'm going to start with that one down here on the right. That is dense regular connective tissue. There's also dense irregular, but we're not learning that, so that's why it says regular if you're curious. Dense regular connective tissue, even if you can't tell this is a bunch of cells smashed together, it's easy to identify because it's just these kind of blurry waves, right? So blurry waves, think dense regular connective tissue, or just dense connective tissue, since you don't need to know the irregular kind. Okay, so that's out of the way. Now these other 3. These are all the loose connective tissues. So you can tell the cells are a little bit more spread apart than here, where they're basically layers of smashed cells together. Areolar connective tissue has the most space between cells. So all these little tiny pink dots, ignore all the hair, the fragment, the hair-looking fibers here. All these little tiny pink dots are cells. So they're really spread apart. There's a lot of white space in between them. And those fibers are kind of, they almost look like just a bunch of hairs on the slide. So that's a good indicator. If you see cells that are really far apart from each other with a bunch of this kind of dyed hair on the slide almost, you know that's areolar connective tissue. Don't worry about what the fibers are. We'll talk about those in lecture. But they're mostly proteins, protein fibers. Adipose connective tissue is fatty tissue. So what you're seeing here, these big white blobs are the cell. And in particular, the white part is lipid, it's fat. So it's a big fat globule in the cell. And you will notice you can't really see any nuclei, right? A lot of times we can see nuclei in the cells, especially this when you're zoomed in this close. This little dark pink part up here on the edge of the cell is actually the nucleus. And you can't see it on every cell. You can see some of them here, up there, But the fat deposit is essentially smashing the nucleus up against the plasma membrane of the cell. So the fat deposit takes up most of the cell. So the nucleus is basically just up along the edge here. So that is adipose connective tissue, and you can see some fibers kind of in between the cells still. And then reticular. So the reticular connective tissue, still a good bit of space between cells. They're not really packed together. And you have these really thick fibers. That's a good indicator that it's reticular. Up here, these fibers are very thin, and the cells are spread out a bit more. Down here, really thick fibers. All right, hopefully that's helpful for distinguishing those. I know they can, especially areolar and reticular, can look really similar a lot of times. But if you look for those thick fibers versus thin, you should be good. All right, so that's our connective tissue proper. Now, cartilage. And I'm going too fast here. 3 types of cartilage. And the difference really is what is surrounding the cells. So the matrix around the cells. Again, the cells are kind of spread out from each other, and some more so than others. But all of these little kind of white things with the purple nucleus in the middle, those are all cells, individual cells of cartilage. They're called chondrocytes. So chondro refers to cartilage, cyte just means cell. And then in between all of that, you have this matrix, which is kind of a liquidy broth kind of thing with some fibers in it. And this type of cartilage, hyaline cartilage, it's a fairly amorphous matrix, so you can't really see any fibers. It's just this kind of blurry backdrop almost for these cells. So hyaline cartilage, amorphous matrix. At the most, you see a little bit of shading here and there. So some kind of shading in the middle, but no fibers, really. Fibrocartilage, you do start to see some fibers. So here's the cells, and then you can see some streaks here, which are collagen fibers. And then finally, elastic cartilage also has fibers in the matrix. So here you have your cells. This is zoomed in a bit more than the other two. And you have these really darkly stained fibers, which are elastic fibers. So a helpful hint, even if you don't really remember, the different types here, elastic cartilage usually stains pretty dark like this. So the elastic fibers hold on to a lot of stain. So you'll see dark staining in cells or in slides that have elastic cartilage tissue. And this may look similar, not the color necessarily, but it may look similar to adipose connective tissue to you. I know when I first learned this, I was like, that kind of looks like the fatty tissue. But the difference there, distinguishing elastic cartilage from the adipose connective tissue, you can see the nuclei in at least some of these cells. And remember, in the adipose tissue, the fat globule basically smashes the nucleus off to the side so you can't see it. So that's how I distinguish, if I'm questioning, that's how I distinguish between elastic cartilage and adipose tissue. So that's most of the hard stuff. I know it's a lot. I'm sorry, I hope you don't feel too overwhelmed, but you'll see it again in lecture, so you'll see it multiple times for this reason in particular, because it's tough to grasp. And then finally, we have bone tissue or osseous tissue. So this is our final connective tissue that we'll talk about. It's easy to identify. To me, it looks like if you cut down a tree and looked at the stump, like the cross section of the stump, that's exactly what bone tissue looks like to me. So you see this central canal. So you have what are called osteons. So you should be familiar with that term in lab. An osteon is basically a bunch of concentric circles. So it'd be like one of those tree trunks. Looking at the cross section of a tree trunk. So this is one osteon, and that contains a number of osteocytes, which are the bone cells. So a lot of osteocytes kind of in concentric circles. In the very middle of that, you have an opening called the central canal or the haversian canal. And that's where blood vessels and nerves actually enter the bones or the bone tissue. And that's all you need to know for bone tissue. So know osteon and then osteocyte as well as central canal or haversian canal. Good. All right, muscle tissue. There's 3 kinds of muscle tissue and then just 1 kind of nervous tissue, so we are almost done. This type of muscle tissue, skeletal muscle tissue, skeletal muscles are the muscles that you think of when you think of muscles. Your biceps, your triceps, your pectorals, your hamstrings, all of those muscles are your skeletal muscles. So this is the tissue that makes those up. This is pretty easy to identify because you have really obvious stripes, which are called striations on these cells. And this is actually all like one line of this. This is one cell. So the cells are really long. There's no divisions between them. And they have a bunch of nuclei in one cell. So multiple nuclei per cell. That is skeletal muscle tissue. And the striations, I guess I should mention this. The striations are basically what allow your muscles to contract. So they're the contractile pieces of your muscles. Skeletal muscle, cardiac muscle is the next one. It's kind of similar, but a few easy to pick out differences between cardiac and skeletal muscle. Cardiac muscle is only in the heart. It's the only place it's found. It has striations. I don't think they're as obvious as the skeletal muscle striations, so they're a little bit less obvious, but they're still present. And then you also have divisions between cells that are really obvious. So these vertical bold lines are borders between cells, between individual cells, which you don't really see in slides of skeletal muscle. And you can see the nuclei very well. And also in cardiac muscle, something I didn't write down here, but a lot of times you'll see branching. So this is one cell. Here's the nucleus. And this is the end on that top branch. And then there's also a little branch down here, and the end of the cell is here. So you'll have some branching of cells, which is kind of weird. You don't really see branched cells in a lot of other tissue types, or really in any others that we talked about. So that's cardiac muscle tissue. Smooth muscle tissue has no striations, so no stripes. This is the type of muscle that's found in your digestive tract and other areas that is involved in kind of slow contractions. So your digestive tract slowly contracts to move food through it. So it doesn't need to contract nearly as often as the other 2 types. Hence, it doesn't have those striations. And I don't know that you would confuse this with any of the other tissues. The indicator here that it's muscle tissue is just that there's kind of these lines of tissue layered on top of one another. And you can see the nuclei scattered throughout, but you don't really see divisions between the cells or anything. Okay, smooth muscle, and finally, nervous tissue, the easy one. The main cell in nervous tissue is called a neuron. All right, so that's it. That's all you guys need for today.